Great. So I'm Dr. Holly Nell. I'm one of, for those who don't know me, I'm one of the PAN-microplastic team members. This is a network that looks to facilitate and encourage knowledge exchange between researchers working on the microplastic field, specifically in Africa. We really want to, I'm just gonna, so yeah, I was just gonna mute you, Tricia, and anyone else, just remember to mute yourself. So this is one of the events that we're going to be holding once a month. So this is the first event, which is basically, so I'm going to keep muting people if there's any background noise. I think Chief, I think you might be unmuted. Thank you, no worries. So once a month, we're going to be hosting these very informal discussion groups. So this is the first one of those discussion groups. And the discussion groups are going to be centered around a specific topic. So today, the topic is back to basics. So we're specifically looking at the sampling techniques and basic analysis and reporting criteria when it comes to microplastics. It's only going to be about a 10-minute talk from my side, and it's not going to be a very lecturing talk. It's really there to give us a point of discussion for the 40 minutes afterwards. So we really want to learn from each other. So please feel free to explain how the challenges of sampling microplastics in your research, in your field, in your river, and what we can learn from your experiences. So I'm going to be sharing my screen now. For those who have just joined, I'm going to link up with the Jamboard again. But I do see lots of people are working on it. Another thing, if you do join, please mute yourself because it is a bit distracting for everyone if there is a bit of background noise. Okay. So I hope you can all see the screen. I can no longer see the chat. So if you have any questions, they will be directed to Elvis will be checking out the chat group. So this is the first club talk, and it's really, like I said, it's going back to basics. And it's looking at the fundamentals of microplastic sampling and basic analysis. Again, I've put this link and I keep going back to it, but I just want to reinforce this is an interactive process. So we have this Jamboard. The link is in the chat. You can see that in the Jamboard, there's a question saying, why do we collect field samples? So why do we collect microplastic samples in the field? And this is going to be a big discussion because this will then feed into how these samples are collected. So what techniques we need to make sure that we're answering the question that we previously are interested in. So how you can contribute to this Jamboard is basically go to the sticky note app. You can click on that and you can add in your comments. So this, the way that I've presented this today is really to focus on papers. This club talk, informal club talk, is based around the idea of journal papers. And although I'm not specifically looking at one article, I did want to bring forward articles that I think are very useful when you are thinking about how to collect samples in the field and analyze them and answer questions that are relevant to you.
Holly, can you maximize your slides, please?
Yeah, are they quite small?
It's just not on presentation view unless it's just me.
Oh no, I didn't put it on presentation view. It's because it makes it, sorry. It makes it difficult for me. I've tried presentation view before and it makes it impossible for me to flip through. So, I think there's quite a few people who aren't muted. If Elvis and Fedora, if you could just go through and make sure that people who are joining the meeting are staying muted, that would be quite helpful. Okay, so the first paper that I wanted to talk about is a paper written by Charlotte Hun, and it's about methods matter. So this, I'm going to give you a brief overview of what this paper talks about, but I will be sending the 3 papers that I talk about today in an email after this presentation. So this paper goes on about looking at methods and why they matter, specifically with microplastics. And it focuses on water samples, but analyzes, I think it's also in San Francisco Bay, and it analyzes and compares 3 different types of water samples. So it looks at manta trawls, it looks at bulk samples, and it looks at pump samples that are filtered, and how these vary according to the size fraction that they collect, the morphology of the microplastics they collect, and the amount of microplastics they collect. And what they find is that 3 different methods show very different concentrations, very different suites of morphologies, and have different issues when it comes to contamination. So this is why they say methods matter, because you need to understand how the chosen method that you have is going to affect the interpretation of your data. So the first thing that they talk about is, why do we actually collect microplastic samples? And that's why I put it as one of the first questions in the Jamboard. So think about your own research and think about, what is it that you're trying to answer? Is it how we previously looked in the past? Is it explorative? Are we looking at presence versus absence? Is that relevant anymore? Or are we starting to move towards trying to have deeper understanding of what's in the field? More hypothesis-driven studies? Is it for mitigation strategies? Is it, for example, I already saw on the Jamboard, because you want to validate a model? All of these questions are going to affect how you collect your samples and what sample you're going to target, whether it's water samples, sediment samples, or various sizes. So this paper gives some really nice examples, and that's why I hope that you will be able to read it after this. But it says that appropriate sampling methods, you need to look at your appropriate sampling methods that capture an accurate estimate of microplastic particles, and that it is the most relevant to the question that you're trying to answer. So it gives a few examples, and one of the examples that I really liked is that if you're trying to investigate the effect of microplastics on a dominant copepod in an estuary, and you know that the copepod only feeds on particles less than 200 microns, does it make sense then to take a sample with a manta trawl that will only collect microplastics above 300 microns? The other example, which was really nice because it brings up to the why, was recently in America and the UK, and not so much in other parts of the world, like South Africa, but there's been these microbead bans. So if you want to understand how effective and how successful a mitigation strategy or implementation tool has been on the environmental microplastic contamination side, would it make sense to collect microplastics not in the size range of the microbeads? So you would have to go and look into the literature to analyze what size range you're specifically trying to target, and then choose the appropriate method for that. So this paper also goes on to talk about the robustness of your method and quality assurance and quality control. And I'm sure that there's going to be a lot of discussions after this about quality assurance and quality control, and the increased need that scientists have called for this in the last few years, that maybe not necessarily was a huge part of the field 10 years ago. But now we need to know, have field blanks been collected? Is the contamination that we're finding in our samples true contamination, or is it contamination through the use of a pump that has a PVC pipe that may have contaminated the sample with PVC? Is there, for example, a need to wash the sampling net between your sites because contamination is a problem with the plankton net that you've chosen, or the shovel and corer that you use for your sediment samples? Is the technique that you've chosen affecting the variability that you're seeing in your sample? For example, if you take a large amount of sediment, you may be able to get a better idea of the area rather than taking one single small sample. How many replicates have you taken? Can you only take one replicate, or in a certain sites, is it necessary to take more than 3, 5, 10 replicates? Is it a high resolution study or is it a low resolution study? And all of these things are going to have effects on the results, and therefore the interpretation and the conclusions that you can get to. So if you move, so again, while I'm talking, really these are just ideas to spark conversation, but go through the Jamboard, because the next slide on the Jamboard is, what do you need to think about when sampling for microplastics? We know that microplastics are very diverse in sizes, shapes, colors, additives, fillers. All of these are going to have an effect on how they behave in the environment. We know, for example, that fibers stay in the water column for a lot longer and therefore may be distributed further. Does that mean that currently our sampling techniques are not adequately detecting these other particles that maybe fall closer to sources or aren't being collected in the tools that we're using? So these are the kind of critical questions that we need to ask. So go into the Jamboard and put your thoughts on, what do we need to think about when sampling for microplastics? And then the third slide is, what information should we include when reporting on sampling in the field? So this is kind of two sides of the same coin. When you think about what technique you're going to use, you need to think about how you're going to report this so that the readers of your paper are able to reproduce the study, not necessarily the concentration, because we know that microplastics concentrations change in a short space of time and in a short area, and very heterogeneous. But could they repeat your study? Have you put all of the information in that study so that they can interpret your data and interpret your conclusions to the best of their ability? So I've put myself out there a bit, and I've actually given an example for us to discuss, and it's not published yet, so, and it's quite far away from being published, but it is something that I think is nice to have an example to discuss. So at the University of Birmingham, where I'm a research fellow, our group is currently, one of the projects we have is on the Ganges. So this is quite a large collaborative project where a suite of different contaminants and water quality parameters were investigated. And in fact, microplastics was not, the study was not designed for microplastics. They were collecting metals, they're collecting, they're trying out sensors. It's a huge project, and it's got 81 sites along the entire stretch. So that's 2,500 kilometers, and there's 81 sites. So we've got to think about the logistics. This is obviously a collaborative project with partners in India, but the microplastic side of things is being done in the UK. So how much sample can I ask to be collected, and how much is it going to cost for it to be shipped back to the UK? Or if people are traveling, which is what the case was, flying back, how much can they carry? How much time is it going to take? As I mentioned, this was quite a large collaborative project. So at every single one of these sites, there were quite a number of other parameters that were being collected. So one of the first things that the scientist asked me was, how much time is it going to take for me to collect these samples? So I had to think of a way that made sure that this was easy and wasn't going to take a lot of their time. The other thing was that, there was only shore-based sampling, so there was no boat sampling, and that the sites were already pre-selected. So this meant that there were a couple of limitations. We were only able to collect a single replicate from the entire stretch. We were only able to collect small samples, so the sediment samples are less than 250 grams. The water samples were volume reduced, but they were also 100 liters. And again, this is only a once-off sampling campaign, which is post-monsoon, which is going to tell you a very different story to if we collected the samples pre-monsoon. But there's a lot of strengths to this paper. We've got 81 sites where we've collected sediment and water along 2,500 kilometers. It's a large collaborative project, as I mentioned, so there's a range of other parameters that I can use to understand what's driving microplastics, and in these hotspots, what other contaminants may be associated with these microplastics. The other interesting thing was that there were a number of sites that duplicate samples were collected, and there were a number of sites where the sites were actually very close together, and they were on both sides of the bank, so the left bank and the right bank. So there's a couple of tricks that we can do. Oops. Chief, I think you are not muted. If you don't mind muting yourself. I can hear myself. Anyway, so
my network is very bad.
No problem. I know. I think you're going in and out. This will be recorded, so we'll be able to share this with you if, after the call as well. So I'm just gonna carry on with the recommendations. So obviously we're still analyzing these samples and we're still investigating, but you can see that I need to be clear when I write this paper that there are a number of limitations to the samples that we've collected that will affect how we interpret the data and what recommendations we can make. And I think there's a big pushback from reviewers and from editors to make sure that the papers that are being published on microplastics are harmonized and comparable at the end of the day, regardless of what method you used and what extraction protocol is being used. So I'm going to quickly move on because we're running a bit out of time. But the other paper that I wanted to draw your attention to is a paper done by Jennifer Provenster, I think her name is. But they wrote this paper called Proceed with Caution, the Need to Raise the Publication Bar. And if you go into this paper, it really talks about the quality of various papers that are being published and the need for researchers to evolve with quite a rapidly evolving field. I mean, we're all in the sphere of microplastics, and we all know that it is extremely difficult to keep up with how fast things are changing. And this paper really goes into various steps that we need to remember when we are publishing and collecting field data. One of the really nice examples that I wanted to put out here is that they find that we really need to be careful when it comes to how we write our titles and our abstracts. So they give an example of a recent paper that was published that said no, I'm not going to say the area, but it said no microplastics were found. And it was in an area that created a bit of stir with the press because, you know, it was previously thought that this area was heavily contaminated and this paper had found no microplastics. But in fact, it's very different to say no microplastics were found above 300 microns. And it's very different to say no microplastics above 300 microns were found within a certain copepod. So what they're trying to show here is that we need to be careful when we're publishing microplastic data, because we are under a lot of scrutiny at the moment, to make sure that what we say has been clear about the limitations, has been clear about validation methods, been clear about reporting standards, reproducibility, and about the really the issues that could go along with it, so that we can have a clear recommendation or conclusion and interpretation of the results. So I'm not going to go too much into the analysis pipeline because I think we've only got an hour, and we're going to have a lot of speakers throughout the year that are going to specifically talk about techniques that help target certain, whether it's a technique such as now red, or whether it's pyrolysis GCMS. So I'm here just laying the foundation of what we're going to be speaking about for the rest of the year. But think about, on this Jamboard, think about what other things we need to take into consideration when we're extracting and analyzing microplastics. So how often do we have to validate our methods? It's quite difficult to validate your method every single time you go through a different sediment sample or field study. But is there a need to do that? We know that if you take sediment samples or samples from the ocean and validate the extraction efficacy is going to be very different to, for example, sewage sludge or river sediments that is heavily contaminated with wood that makes it difficult to digest. So these are things we need to figure out the trade-offs of how often we need to validate our methods and how often we need to run these positive and negative controls. And then again, what information do we need in our reporting standards to make sure that the community that's reading our papers can reproduce our study? One of the big things that a lot of people are having conversations about is you want your study to be included in these syntheses or meta-analysis studies. You don't want it to be excluded because you weren't clear about the limitations and someone can't trust your data. So although we can get a lot of information from these site-specific papers, it's really these broad synthesis studies that are going to show how we progress and how we move and how we push the boundaries of this field. So just lastly, there's another paper, this is the third paper that I'll send out, called Reporting Guidelines. And I find this quite useful when I'm writing a paper, because in the supplementary, it actually has a list of things that should be included when writing up your paper. So what should be included when you're writing about your field sampling sites? What should be included when you talk about the extraction of your microplastics from a certain technique or the visualization? And it's a really good checklist for students or for, I mean, I'm not a student anymore, but I still use this every time I write a paper to make sure that I am including, because it's easy to forget because there's so many steps, I'm including everything that will help make my data interpreted in a certain way or reproducible at the end of the day. And the two things that I find really useful is making a lab-based pipeline. These change for every study, and they can often be put in the manuscript, or a lot of people put them in the supplementary, but they're very, very useful for yourself and for the reader to interpret exactly what steps you took to extract those samples and to get to the data that you got to the end of the day. And you can see on the right-hand side, and I will share these slides with you, where you can then go back and interrogate your own work. So I can say, okay, well, I took a bulk sample, but actually my bulk sample was rather small, and maybe the larger particles are being underrepresented, and that it's better for particles less than 1 millimeter. And this is going to affect how we analyze and interpret our data. For example, we use sediment microplastic isolation units, and we have tested the extraction efficacy, but maybe I need to test the extraction efficacy for every study, which we don't do at the moment. Maybe we have observer bias or user bias. If you're working with a lot of students, there's a lot of training involved, and maybe one student, for no fault of their own, is better or worse at identifying microplastics. The other thing is, for example, now red, should I stain or should I not stain? So there's a lot of questions that I think you need to constantly be asking yourself if you want to improve and make everything reproducible. And the last thing is also these identification keys are very useful, and you see them in a lot of papers. It really shows the reader exactly why you chose that particle as a plastic and why you discarded another particle as a non-plastic. So I'm going to stop there because I know I'm going a bit over time and I really want to open it up to everyone else. So please feel free to ask questions to myself, but also just introduce yourself and tell us what difficulties you've had collecting microplastics in the field, or any advice that you have for something that has helped you overcome a problem in the field or even in the lab. I don't see anyone's hands up. And I don't know if I can see anyone on the chat group. Oh, Conrad. Conrad, I'm going to unmute you. Yeah, I think you can. Hey.
Hi, everyone. Thank you very much, Holly, for a very interesting conversation. I think it's a great start for us to start chatting about what we're doing. I'm Conrad Sparks. I'm from the Cape Peninsula University of Technology, all the way down south, South of Africa. We started doing microplastics research about a year or two ago. And Holly, one question I have, which I don't really see being part of the reporting of publications, is the verification of microscope counts. And I found that with a student where I trained the student, I told her what to do. I then made the assumption the counts were correct. She then finished it was for a research project, but I want to publish it. And then I counted the samples myself. And I counted a third of what she had counted. And since then, I've asked the students to validate each other's work. And I would come in and do a random check of the samples. And if we way off, unfortunately, the student will have to start again. So I encourage them to check each other's work, to validate each other's work before I do a random check. But I just don't know your thoughts in terms of that step of the process. Thanks.
No, I think, yeah, that's a very, and I know it was a whirlwind of me throwing a whole bunch of things on slides, and there were so many things that I left out. And I think that's a really important point that you've made there, especially because a lot of us are working with students, and it's impossible, even if you're not a student, to be able to, one day you go and count and then the next day you're slightly different. I think it is very difficult for all of us working in the microplastic realm. There are a number of things that, yeah, I think there's a number of things that you can do. For example, have multiple counts. This identification key is also very useful for students to make these identification keys before they start and stick to it. Because what you often find with students and with myself is halfway through, you're going through a sample, you start doubting yourself. You've been sitting with the microscope for 8 hours a day and you start thinking, oh, am I actually counting the right things here? So having these identification keys and sticking with it is very useful. And the other thing is that I recently went to a meeting about method development. And one of the things that will eventually come into place are these I don't like the word tests, but they're going to be proficient quality control tests where an external lab is going to be sending out unknown amount of microplastics in a sample to various labs, and you can then get a certificate to say that you're able to count them. So it's good that we're thinking about these things now on how to quality control ourselves, because eventually there may be a time where we're going to be externally audited when it comes to this as a contamination, especially when our results are affecting policy and things like that. So it is a very good question, and it's something that we need to think about in our labs. And we can also send each other things, Conrad. You can send some stuff to our students, and we'll send stuff to your students and see if we come up with the same number.
A great idea. Thanks. I think we can chat about that. Thanks.
Anyone else?
Only there is a question in the chat, so maybe if you can unmute Gideon Idowu to just ask it. I don't know if Gideon.
I can't find, there's so many people. I can't find Gideon, but Gideon, if you're there, if you want to unmute yourself, or we can ask the question on your behalf.
Yeah, hello.
Yeah.
Can you hear me?
Yeah.
Yeah, we can hear you.
Okay, I just have 2 quick questions to ask. The first is it compulsory to digest samples after extraction of the eliminate other non-plastic materials prior to estimating the amount of microplastics? That's the first question. And then the second question is whether it is important to do scanning electron microscope SEM as part of microplastics analysis.
So,
did you get my question?
I did. Thank you, Gideon. So yeah, these are really good questions, and this is what this platform is. This platform is about. I guess with the digestion step, we do digest every sample that we have, purely because we work with NileRed, and NileRed can have issues with false positives, so it can stain organic matter. So we try to remove as much of that organic matter as possible. And I think getting rid of as much of the organic matter as possible is also good when it comes to things like FTIR and ramen. But I don't think it's always necessary in every case. I think there's going to be certain, like sludge and wastewater treatment samples are going to need more digestion than maybe a water sample. Or a water bottle sample. I know that, Fedora, you recently looked at samples in drinking water, and maybe that doesn't necessarily need as much digestion as, for example, a water sample that is heavily contaminated with other organics. When it comes to SEM, I don't know if anyone in the audience wants to answer this question. I personally don't use SEM for any of our microplastic work. But we're not specifically looking at the structure and shape of the microplastics. We do use SEM for more ecotoxicology and things like that, when we want to know the shape and the exterior of the particle. But I don't know if anyone else wants to raise their hand if they use SEM at all. Andrew, I think you're unmuted.
Yeah, I can maybe comment. Sorry, I can't find a raise hand command on my Zoom screen at the moment. It seems to have disappeared from my reactions box. But yeah, just a comment about SEM. It's a really useful technique if you want to know about the detailed morphology and surface properties of polymers. If you're interested maybe in weathering properties, things becoming brittle and fragmented, all these kinds of questions. But as a routine technique for microplastic identification, I don't think it's really very useful because, well, it depends what kind of SEM you have, of course, but even if you've got EDX capability and so forth, unless you have heavier atoms in your polymers, you're not likely to find all that much that's really helpful to you. You know, sulfur in tire residues, maybe something you could pick up by EDX. But a lot of very common polymers like polyethylene, polypropylene, PET and so forth, they don't really have any atomic constituents that are easy to pick up by SEM. So you can't really do very much verification that what you're looking at is really a plastic. So you need to do that other ways and then use the SEM if it's useful to help you to understand something about the particular properties of those particular microplastic particles.
I see there's another question about these identification keys. And I think as Charles asked us, how can we obtain these microplastic identification keys? And it's an interesting question because I think we make our own specific for our techniques. So we've validated our techniques, and we then make an identification key for the different studies. So each study has a different identification key, and that's going to be different to, for example, the identification keys that maybe Andrew would use, or you know, Fedora uses, or even Elvis, who uses pyrolysis GCMS. This is also a great platform maybe in the future for us to have maybe a shared document where we can put pictures of microplastics. I think it's very useful for students to look at all of the different microplastic pictures that have been taken from researchers around the world and use those to create their own identification key in a way. So maybe this is the platform for that. I see Hendrik, your hand is up. I don't know if you want to unmute yourself and ask that question.
Well, it's not specifically to that question, but pertaining to it. You're closer to the heat. Do you have any information on if there's any movement towards standardized ISO sort of procedures, or you know, any of those formal EU procedures, processes? Is there any movement towards that?
I think I'll quickly go, and if there's anyone else that wants to pipe in here, but there's a lot of discussion about standardization versus harmonization, and the difficulties that we're facing in comparing our data with each other and things like that. And there's a lot of school of thoughts where I think a lot of people think that we are definitely closer to getting a standardized set of techniques, where there's other people who think it's never going to be a case, and actually we need to work more on reporting standards and making sure that we're clear about the limitations of the different methods that we have chosen for our specific study and for our specific question. When it comes to those standardized methods, I think maybe and that's not really where we focused here, in toxicology and health and food, I think they're pushing more for standardization than maybe in sediment and water and things like that. But I could be wrong here. I don't know if anyone else wants to bring Fedora. I don't know if you.
Yeah. Good afternoon, everyone. Can you hear me? Hello?
They sound good?
Hello?
We can hear you, Fedora.
Oh, great. Yeah, good afternoon, everyone. I just want to comment on digestion one, then the SCM. On the digestion, I think Oli has mentioned that it depends on the sample. And I think Tristan is making a comment in the group that I think everyone needs to be aware of, that it depends on the sample. Some polymers do decompose with digestion. I remember Elvis had a review on different digestion methods. I don't know Elvis, if I'm right. Yeah, I think he had a review on that. So we need to be careful on the kind of the sample we have before we undergo digestion to make sure that we do not underestimate, the counts at the end of the day. Andrews has also talked about SEM, but it's also important to point out that presently there are some studies that are going on that some metals are adsorbed on the surface of microplastics. And SEM EDS can actually give information on this. And I think there are some toxicological studies that are talking about the co-exposure of microplastics and heavy metals. Yeah, and I think understanding how heavy metals interact with microplastics, SEM EDS is useful in analyzing that. So, and also a lot of studies are still going on to understand how SEM can be used to improve the information that are extracted from microplastics. And I think anyone can also venture into that, trying to understand what other information, apart from the morphology, and the surface texture, what other informations can we get from SEM? Just like we know that pyrolysis GCMS is an imagining method. So we can actually sit down and also see how these instruments can be used or effectively used or in combination with other equipment to be able to get good information from our samples. Thank you.
Great, thank you, Vidara. I don't know if anyone else has any questions, just to spur on a few things with this idea of standardization and harmonization, and it's something that I think I've thought about quite a bit. Is there's another paper that talks about the microplastics as a suite of contaminants and how diverse and complicated they are. And I think it's important that and hopefully we will have this discussion with other researchers who work on maybe larger particles, that just because we're not always detecting the smallest particle is not a bad thing. If we, if your question is to, like I said, with the microbeads or nurdles or things like that, if your question is to understand how Operation Clean Sweep, which looks at mitigating the spillage of nurdles in the environment and how that mitigation strategy is improving pellets in the environment, then I think there are quite standardized methods. I think the standardization gets more difficult as the particles get smaller, because there's so many methods coming out. But for particles above 500 microns, I think things are fairly standard from what I can gather.
Yeah, or maybe let me contribute to the issue of standardization. And I think this is one of the reasons why we are having different small groups, and I think it's important that once we have a good documentation of our procedure and we follow a good quality control, as we bring together ideas from every part of the world, those are procedures that are actually well documented. We actually form part of the standards that is eventually going to be accepted globally. So, and that's why we said that it's important that when we are making our documentation, we should be thorough enough in order to show how we have gone about our experiments. But I think at the global level presently, people are still discussing on standardization. And our reports, our publications will determine what it's going to look like. And that's why we really need to give details of whatever we are doing in our various labs. And that can help us to get a good picture of a good procedure at the end of the day. Yeah, thank you.
Thanks, Vidara. I think we've also got another question in the chat, and I'm going to try to pronounce the name, so I do apologize if I'm completely way off, but Umumi. Yeah. I don't know if you would like to unmute yourself and ask a question, but I'm also happy to ask the question on your behalf.
Hello.
Hello.
Hi. I just want to know if there are, because I know that counting microplastics is quite tedious. And the smaller the size gets, the more difficult it is to count. So I wanted to know if there are instrumental techniques available for counting, especially for very small microplastic particles.
Yeah, that is that is a very good question, and I think that's something that we're going to tackle in the next few club talks. So I'm going to answer the question and bring it to everyone else, but it is something that we will look into more, because there are a range of different techniques. I know Andrew Mace is here, and he works on now red and imaging. And this works quite well for particles. I think it's 10 microns, which is your lowest limit, but I could be wrong there. But then there's also Elvis, who works on pyrolysis GCMS, where you put the entire sample in and you can get readings, which is good for smaller particles. And then I don't know if we've got anyone here that works on FTIR imaging. I think, again, sorry, Andrew, I keep putting you on the spot, but I know that's also a technique that you use. So maybe, Andrew, if anyone else wants to also have a stab at that.
I can have a quick go at commenting on the imaging with Nile Red. And there are automated techniques you can apply to that. And the resolution of the technique just depends on the optical system you have available. So depending on the magnification of your microscope or your photography system or whatever you're using, that will dictate the size of particles that you can detect. But in theory, you can detect single photons from fluorescence. So you could even detect nanoparticles via Nile Red staining if you had the right optical configuration. Whether you could verify they're actually microplastics or nanoplastics or not is an entirely different question. But purely from the point of view of counting them, you can certainly do that. The problem is that all automated particle recognition techniques are, in my opinion, quite unreliable. They all work fine on nice clean test samples with nice spheres or nice discrete particles. But when you get a really complex real sample and you add to that the fact that very often your staining is uneven and some things are brighter than others, it becomes very difficult to automate these counting processes. And there are a number of publications, for instance, that use ImageJ, which is great because it's free and it's a very powerful piece of software. But the problem with if you use ImageJ is you basically set a background threshold and you say anything brighter than this line is a particle. And the problem is you can make the number any number you want it to be, depending on where you put that line. So it's, in a way, it's objective, but how you calibrate it in the first place is subjective. And this problem hasn't really been very effectively solved at the moment. There are a number of groups working on much more sophisticated solutions that use, better image analysis algorithms that look at how bright a particle, an object is relative to the things immediately around it, rather than looking at a global average for the entire field of view. So that is probably a very helpful approach. I'm sure people will come up with some very clever AI-based techniques to eliminate certain types of objects which are false positives or which can be identified as organisms because of their shape and characteristics. So I think over the next 2 or 3 years, probably, quite a lot of publications will come out in this area that will deal with some of these issues. But at the moment, I would have to say, based on our own practice, we mostly rely on our eyes at the end of the day. Because although they're subjective, they are extremely clever at dealing with issues of uneven lighting and things like this, that we as humans, we can understand what we're looking at, and we can assess what we're looking at in that context, whereas the software that's currently being used doesn't really do that.
Yeah, I think Andrew's hit the nail on the head there. I think there are a few papers that there's a lot of lovely techniques out there, and I can't wait to get my hands on them and practice with them. But if you actually go into the literature, a lot of the techniques also have their limitations. If you're imaging with FTIR, a lot of the time they don't pick up fibers at all because of how thin they are, and they only pick up fragments. And we know that fibers can be quite dominant in the environment. So I think that's what today's talk was about, was really everything has its limitations, and we need to be clear about those limitations, and therefore interpret our results as accurately as possible for the reader. So I know that I don't want to keep everyone for too long, but if there's any burning questions, we can
just give a comment on the size, the smaller size analysis. So in our lab, I'm Elvis, part of the Pan-African microplastic team. I analyze plastic using pyrolysis GCMS. And to be honest, I mean, when it comes to nanoparticles, smaller size particles, I think currently pyrolysis seems to be on top. It has its limitations, like Police rightly pointed out. So what we do in our lab is that when you do the digestion of the samples, and then depending on which sizes you want to analyze, you can actually filter them onto different size filters. And then in that way, you can analyze the filter, which will then give you the concentration or the amount of plastics in that size range. So pyrolysis is very good in that sense. We're able to get the nano size plastics. And I think, like Holly said, I think FTL is not able to go below 20 micron. If I'm sure, I've seen people saying that in literature, and I've seen literature saying that Raman spectroscopy can also not go below, I think, 1. And I think I'm doing a study in the lab currently, and I found that all the studies that are actually quantifying plastics in biosolid are actually underestimating, I think, about 31% of plastics in the samples, because I picked out the lowest sieve that people have been using, and all the literature data that I have, they all counted plastic above 25 micron. So we decided to then try and analyze below the 25 micron. And then we found out that, like, if people quantify, the current studies are underestimating like 30% of the amount of plastic that's supposed to be in the sample. The data is still coming out. Hopefully we'll get it out and people can have a read. So pyrolysis seems to be good. But like I said, it's still, you have to still go through the sample extraction, filter them onto a filter paper, depending on how or which size you want.
Great, thank you, Elvis. And Elvis will also be presenting at one of our club talks in the future. So I'm going to wrap up here. Thank you so much for joining today, and I hope it was informative. These are really meant to be informal discussions, and I know we can't answer all the questions all in 1 hour, but we are going to be having these on the first Thursday of every month, and these conversations will progressively start answering the questions that we have throughout the process, from extraction to analysis to interpretation. If you at any point are interested in presenting or being a moderator and being on my side, where you give a bit of a presentation and have a discussion around a certain topic, whether it's modeling or pyrolysis, GCMS or something like that, please do just get in touch with us. I do think everyone's got my email address, and I will be emailing everyone the recording and the 3 papers and presentations and the Jamboard things. So yes, do get in touch, and thank you very much.